Okay, good morning, students. Today we're gonna learn four learning objectives. Okay, first we're gonna look at explain that stomata respond to changes in environmental conditions by opening and closing, and that regulation of stomata aperture balances the need for carbon dioxide uptake. So carbon dioxide uptake by diffusion, this is photosynthesis, and the need to minimize water loss by respiration. The second one is explain that stomata have daily rhythms, opening and closing, and then the function of structure of and function of gut cells, mechanism of opening and closing of stomata, and the last one is on abscisic acid. Okay, describe the role of abscisic acid in the closure of stomata during water stress time. In other words, when water is not or water is scarce, water is not present in a huge amount. Okay, so first of all, I think I've shown you this slide before, so this is just this is also a recap. I think I remember teaching you this, so it's okay. This is if I have taught you before, then this is a revision. Okay, so basically stomata naturally are found more in the lower epidermis compared to in the upper epidermis. So more in the lower and less in the upper epidermis, and this is to prevent excessive water loss. Yeah, and then these are just some data just to prove that the lower epidermis, see 6,000, 5,000. There are some cases where the lower epidermis has less stomata, but when you look at 29,000, 0, 28,000, 4, so majority, as you can see, there are more more stomata on the lower epidermis, yeah, compared to the upper epidermis. Okay, so this is a stoma, yeah, which is closed, yeah, a closed stoma. So stoma, actually, the actual meaning of stoma is whole, yeah, whole, created by two gut cells. And this is actually one gut cells and another gut cell. So gut cell is actually an epidermal cell. Okay, it is a special type of epidermal cell. In this case, it's called gut cell. Okay, these are just some images. So stoma versus stomata. So strictly speaking, so a stoma, it means whole between the gut cells. But the term is usually referred to the two gut cells and the hole between them. Okay, so when we looked at like for example this one here, so a stoma normally nowadays, generally speaking, it means this one here. This whole thing here is stoma. But strictly speaking, stoma means the whole. Strictly speaking. But in general nowadays, when we say stoma, it means the whole together with the gut cells. Okay. Okay, let's have a look. Now we move on to daily rhythms of opening and closing of stomata. Now this is pretty interesting, yeah. This is taken from your textbook, yeah. So if we look at the during normal day, so when there is sunlight, so this is as you can see, 12 noon. Okay, this is when light intensity is the highest light intensity. So when the presence of light will cause the stomata to open, because it's gonna let carbon dioxide in. Okay, carbon dioxide in for photosynthesis. Okay, so when there is light, the plants will photosynthesize, It requires carbon dioxide. It requires carbon dioxide, so the stomata would need to be open, yeah, to allow the carbon dioxide to enter and then to release the oxygen. Now, you have to note as well. You have to note that when the stomata are open, okay, especially during this photosynthesis, yeah, water vapor, yeah, in the gaseous state will also escape at the same time. Yeah, water vapor in its gaseous state, water in its gaseous state will escape at the same time. So transpiration will occur at the same time. So this is the meaning of inevitable consequence. Inevitable consequence. So transpiration or the escape of H2O gaseous state cannot be prevented. Yeah, transpiration will occur if the stomata are open. Why are the stomata open? Because they're gonna let carbon dioxide in for photosynthesis. So but there's no problem in there shouldn't be any problem here. So even though the water vapor escape, if the water if water replacement is higher than water vapor escape, then it should be all right. Yeah, in other words, if the water supply is greater or water availability is greater, water supply is greater than the water vapor escape, then there shouldn't be any problem. Okay, but it will be a problem if water supply is less compared to the transpiration rate. Then if that's the case, then the stomata would need to be closed, yeah, to prevent excessive water loss. Okay. Now what else can we take note from this diagram here? As you can see, if you look thoroughly on the y-axis, so the stomata are not completely closed. Yeah, stomata are not completely closed. Yeah, even though when this dark area, yeah, dark, see from here, darkness. So when there is no light, you see the stomata is not 100% closed. Yeah, the aperture is still slightly open. It's not completely zero, and this is to allow respiration to occur. Yeah, respiration occurs all the time. Respiration does not stop. Yeah. Of course, the rate of respiration is lower in the absence of light, yeah, as compared to when light is present. Okay. The second thing to note is, so when this is normal light. Normal light means normal day and night, meaning there is a peak in the light intensity, and then the light intensity will gradually decrease at its afternoon and then evening and then night time. Yeah, but if you were to do an experiment and if you were to do it by giving constant light, meaning the light is constant, now you can see quite amazing results here. Yeah, you can see when light is given constant, it has the same rhythm. The rhythm is the same. You see, there's a peak here, peak, and then there's a downhill here, peak again. Okay, as you can see, the pattern looks the patterns look very similar. Yeah, it looks very similar. Yeah, even look at this one here. Yeah, they look very similar. So what does this tell you? Even during constant light, yeah, let's say during constant light, yeah, the stomata is also closed, yeah, virtually closed, yeah. um, even if when light is present. So this simply means that there is something that which is known as internal clock. Internal clock. So these plants, they have like an internal clock which is already set. Yeah, that at a certain time the stomata will close even though there is light intensity. Even though there is light intensity, at a certain time the stomata will close. So they have what we call this internal clock. That is the reason why um, uh, rhythm, it's called rhythm, daily rhythm. The rhythm is almost constant, even when you long periods of darkness. So when it's exposed to long period of darkness, uh, so this is during darkness. You see, when in long periods of darkness, the stomata are open. This part here, this area here, this is the long period of darkness. Okay, so as you can see, it still has that decrease, the same as this one, then increase, and then decrease, and then increase. Yeah, but maybe this is it's not applicable because not darkness. This part here, you see, and then sort of increase and then decrease again. Okay, so you can see, even if you expose these plants to darkness, there is still they still responds by opening the stomata, especially this area here and this area here. Yeah, why do these plants still open the stomata despite that they're exposed to darkness? No light means there's no photosynthesis. Yeah, that is because they have that what we call again internal clock. Okay, internal clock. They have internal clock. Okay, but of course it is not as big. For example, it is not as the opening of the stomata is not as wide as when there is constant light. Yeah, this is a long period of darkness. So but they do still respond, they do still have follow their daily rhythm, that internal clock. Okay, let's look at the next one. Stomata respond to changes in environmental conditions. They open. So of course, when there is light intensity, they will open. The stomata will open because this is to allow carbon dioxide to enter. Okay. And when there is low carbon dioxide concentration in the air space, so when there is low carbon dioxide concentration in the air space, the stomata will also open so that more carbon dioxide can enter. Okay. Stomata are open, leaves gain carbon dioxide for photosynthesis but tend to lose much water in transpiration, inevitable consequence of stomata opening. So this is again what I will mention again. Whenever stomata are open to allow carbon dioxide to enter, transpiration will occur at the same time. This cannot be prevented. Okay, this is inevitable, meaning it cannot be prevented. Transpiration will occur. Water in its gaseous state will escape. Okay, but this is still not a problem. Like I said, provided that water supply is higher than water loss. Yeah, water supply is higher than water loss, the water vapor loss, okay, to the atmosphere. Right, next one. Stomata close in response to darkness. Okay, in response to darkness, um, we can say that yes, in general. Yeah, in general. But if you look at this one again, um, but it's fairly the opening of the stomata decreases, yeah, in darkness. Okay, even like this one here as well, as you can see. Yeah, in darkness, yes, stomata they they respond, yeah, um, by closing the aperture. But in this experiment, this is just to show you that it's not completely closed, yeah, because they have that internal clock inside them. Yeah, they also follow the internal clock. Okay, yeah, but in general, yeah, stomata close in response to darkness. High concentration of carbon dioxide in the leaf, so there's no need to open the stomata. For example, so if there's a high concentration of carbon dioxide here, okay, so if there's a high CO2 here, so this stomata will close, yeah, because there's no need to open the stomata because it has lots of carbon dioxide already, yeah, and this is to prevent transpiration, yeah, to prevent um, transpiration, to prevent excessive water loss, yeah, the loss of water vapor. Low humidity. Um, Low humidity means low concentration of water vapor. Okay, outside. High temperature. Water stress. Okay, because low humidity means low humidity again when we or high temperature. These are all related to low humidity, high temperature. This is all related to um, transpiration. Leads to increased transpiration rate. Yeah. So low humidity and temperature will lead to increase in transpiration rate, and that's why the stomata will have to be closed. Will have to be closed. Yeah. Um, because otherwise, then the plants will lose too much water vapor. Okay. So in times when conditions which can lead to high transpiration rate, the stomata will close. Low humidity outside, this will increase transpiration rate. High temperature as well will also increase transpiration rate. Water stress, yeah, this is going to talk about this later on in more detail. This is on the abscisic acid. Water stress, that is when availability of water in the root is limited, when there are high rates of transpiration. Okay. All right. The mechanism of stomata, all right, is that it looks something like this. Yeah. So you have to understand that this is thicker inner wall. This is inner wall. Thicker. So if it's thicker, it means less flexible. Thicker means less flexible. And this is the outer wall. So outer wall. So that means thinner, more flexible. Okay. So the same thing as this one here. This is the inner wall. This is also the outer wall. Okay. So when water enters, H2O. So when H2O enters here, okay. So only this area will expand. This will expand more. Yeah. This area here will expand less. Okay. So eventually you can see that the shape will be like this. Yeah, if water enters. So you have to learn the structure of the stomata as well. The thicker inner wall, this is the inner wall, and then the thinner outer wall. And then this is more flexible, and this is less flexible as stated here. Okay. All right, the mechanism is very, very important here. The what causes the gut cells to become flaccid? So this is from close to open, yeah. So from close to open. Uh, so as you can see here, the pumping of protons occur. Okay. This is a different. So the first thing is the pumping of protons. Okay. So there is a pump routine here. Of course, ATP is needed, and of course the process is active transport. Okay, so the pumping of this proton um, to outside of the gut cell. So outside means these are epidermal, yeah, epidermal cells. You have to imagine that these are epidermal cells, yeah, epidermal cells. So the pumping of this proton is out of the gut cell to the epidermal, yeah, epidermal cells, will cause potassium channels to open. So potassium will enter. So this is facilitated diffusion, facilitated diffusion, yeah, facilitated diffusion, because high concentration, low concentration. Okay, so so the entry of potassium here will cause high solute concentration, and then of course low water potential inside the gut cell, yeah. So then water can now enter. So when water enters, that's when it creates the stoma, the hole. Okay, so it's very easy, yeah? You have to just think that in order for the stoma, in order to create the stoma, which is the hole, yeah, water has to enter, yeah, meaning low water potential inside, high water potential outside. So in order to create low water potential inside, there has to be high solute concentration inside. High solute concentration. So think in that manner. How can we make high solute concentration? And that is by allowing potassium to enter. And that can only happen after the pumping of the protons. Okay. Now, once you know from close to open, then it will be easy how to go to how to reverse the process, because once you know the one-way process, then the reverse is very easy. So the reverse would be there's no pumping. There will be no pumping. Okay, no pumping. No pumping of protons. Okay, now no pumping will now lead to potassium to leave. Yeah, to leave the gut cell to the surrounding environment, which is the epidermal. Okay, so again you have to remember that these are all epidermal cells, yeah? Epidermal cells surrounding it. Okay, no pumping means potassium will now leave, because this is high concentration inside, low concentration outside. So that will lead to more eventually. Remember, high, low, eventually there will be high solute outside, which is low water potential, and then eventually low solute inside, which means high water potential. Now, meaning after this one here, after the potassium has left the gut cell, the solute is high in the outside, low water potential. The solute will be low, high water potential, and so water will now leave. So when water will now leave, becomes flaccid. Okay, so it's very, very easy. Okay, and this is the last one. So basically we have covered the uh, structure already, respond, and daily rhythm already, looking at the graph just now. Now the last one is the role of abscisic acid during water stress time. This is the last one, objective number 4. Okay, objective number 4. Um, now, the abscisic acid is a stress hormone. Okay, it's found in every plant. It is a stress hormone. It is released when plants are experiencing tough times, like high temperature, very high temperature, low water supply. Yeah, it is a hormone, um, which will lead to a particular consequence, and that is the closure of the stomata. Now, let me um, so if I were to let me just rotate this. If I were to uh, create, yeah, stomata here, thicker inner walls, thinner outer walls. Yeah. So let's use a different pen. Okay. So abscisic acid, abscisic acid. Okay, ABA will bind to this receptor here. So let me just binds to the receptor of the cell membrane. Okay. So when it binds to the receptor, so there is no protein pump. Sorry, there is no hydrogen pumping. So this hydrogen pumping, low concentration, high concentration, this does not occur. Yeah. Once it binds, it doesn't occur. There's no pumping. Because there is no pumping of this proton, then it opens calcium channels. Let me just use a different. So there's no pumping, so chloride ions will leave. And then potassium channels will also leave, K+. Okay. So because of this action, let me just refer back to my sorry, I forgot about one thing, and that is the calcium. Yeah. So the binding of, let me just see. I missed one thing. So ABA binds the receptor. Proton pumps are inhibited. All right, that one is done. That one is correct. I forgot about one more thing, and that is the second messenger. So there is a second messenger. No binds the cell membrane. Pump protein inhibited. No pumping. Calcium, ABA, stimulate calcium to enter. It acts as a second messenger. Chloride channels leave the gut cell. Potassium channels also leave the gut cells. Okay, let me rephrase again. Yeah. So uh, okay. So abscisic acid, okay, binds the receptor here, prevents the proton pumping, okay, will cause calcium to enter. Now calcium will enter, Ca2 plus, Ca2 plus. This calcium act as a second messenger. Calcium acting as second messenger. Now, what do we mean by this second messenger? So cell signaling. So like I said, because this is a hormone, remember, this is a hormone. The binding of a hormone, normally there is a specific cell signaling process that happens inside the cell. Just like when we talk about glucagon binds to the cell receptor, there is a particular or a specific cell signaling, yeah, ADH as well. The same thing here with abscisic acid, because it is a hormone, so normally there has to be a cell signaling process that occurs, and in this case the calcium is acting as the second messenger. We're going to talk about that later on, okay? And because of this, the calcium will cause chloride ions to leave. So now we can draw the chloride to leave, and then followed by calcium to leave. Sorry, potassium, yeah, chloride ions and potassium ions to leave. So this calcium acting as a second messenger, it causes other reactions and things like calcium to leave, chloride ions to leave the gut cells, and then potassium chloride ion to leave the gut cells, and then potassium to leave the gut cells. So eventually, what will happen inside here? So inside the gut cells will have low solute, yeah, low solute concentration, and then of course high water potential. So water will leave. Yeah. So here water will leave, and so this stoma will be closed. Yeah, the hole will be closed. Okay, so this is stated. Unfortunately, it is stated in the learning objectives. Okay, the role of abscisic acid in the closure of stomata during times, including the role of calcium as a second messenger. Okay, so it's already summarized here, yeah, as a flow chart. Okay, so you can check, yeah, this flow chart here starts from here. So it begins from here and then it ends here. Okay, this is the last one. So you can read that, yeah. Now, just a recap of the cell signaling pathway. So this is the abscisic acid, the hormone, binds to the protein receptor. Okay, complementary shape. So that changes this shape here. Conformational change. Remember, conformational change. What does it mean, conformational change? It changes the shape so that the G protein can bind. Okay, so now the G protein is activated because it has bound to the receptor here, and it will move along this inner membrane, and then it will activate this enzyme. Now this enzyme here, in the case of glucagon, it's called adenylyl cyclase. But here it's not mentioned because the enzyme, because this is a different reaction, so the enzyme is different. And this will cause the making of second messenger. So in this case, the second messenger here is calcium. So in other words, the calcium would need to enter in this case from outside, the entry of calcium. So the entry of calcium will cause other reactions here. The other reactions is the exit of chloride, and then the exit of potassium. Okay, the exit of chloride and the exit of potassium. So it causes everything will cause low solute, high water potential, and so water will leave. So when water leaves, then becomes flaccid, yeah, and the stoma will be closed. Okay, so the process of cell signaling pathway is actually the same, yeah, with the protein receptor. It has a receptor here, but the receptor is different. It's not the GPCR, yeah, G protein coupled receptor. That's for glucagon. This is different. The name is also different. It has the same G protein. The enzyme here is also different. The second messenger is also different. And then what happens, the response is also different. Okay, but it has that same sequence of events in the cell signaling process. Okay, and some of the past paper here, cells have chloroplast, but epidermal cells do not have chloroplast. State other difference visible, gut cell and epidermal cell. So of course, the other difference is the thickness of this inner wall, and then outer wall is thin. Whereas we just use a different color. So yep, inner wall here is thicker, outer wall is thinner. Whereas for epidermal cells, they are the same throughout. The size is the same throughout. Okay, precisely where epidermic acid binds cell membrane of gut cell. Ion that diffuses from gut cells to epidermal cell. This is stomatal closure. Yeah. Identify the ion that diffuses from gut cell to epidermal cell. So this can have more than one answer, yeah. It can be potassium, because potassium, as you can see here, they leave from the gut cell to the epidermal cell. Remember, this is all surrounding epidermal cell. Or it can also be chloride, yeah, chloride. So this can be potassium or chloride. Compare the relative water potential of the gut cells of the epidermal cells. Water potential higher water potential in gut cells than epidermal. Okay, higher, because water will eventually leave, yeah. The change in the volume, so volume decreases as water leaves gut cells, you can say by osmosis. Okay, Okay, this is just one of those past papers which are, important for you to go through, yeah, and try to understand the ion that diffuses from gastroepidermal cell, that can be potassium or chloride, yeah, because both during stomatal closure, especially if it's not related with abscisic acid, if it's not related to abscisic acid, yeah, ion that diffuses from this would be potassium if it's not related to abscisic acid, meaning not during stress time. So not during stress time, then it will be potassium, because it will be this one here. Okay, this one. But if during stress time, meaning during abscisic acid binding, then it can either be potassium or chloride. Yeah, K plus or Cl minus. Okay, right. That's all for today. I think it's slightly beyond 20 minutes, if not 30 minutes. Okay, we'll continue again tomorrow. Yeah, that's all for today.